Hello, my name is Andrew Ribeiro. I'm the head of the Laboratory of Biosystem Dynamics at Tampere University. Today I'm going to talk about our findings that were reported in the scientific article Effects of Sigma Factors Competition Are Promoter Initiation Kinetics Dependent. E. coli has evolved genes to carry out programs under normal conditions, such as processing food. Meanwhile, other genes assist E. coli in responding to stress conditions, which are more rare. In this work, we focus on the dynamics of transcription programs that are activated by sigma factors, which are proteins that, once present in the cell, quickly activate and repress many genes. In detail, when detecting a stress, E. coli cells quickly produce many numbers of a specific sigma factor. This molecule then activates and represses several genes. Interestingly, when this occurs, other genes which are not directly responsive to this sigma factor also tend to have their own activity reduced. In some of these genes, the activity is very reduced, while in others, the activity is only weakly reduced. It is unknown why there is such a diversity of indirect responses. Here, we propose a mechanism that can explain this variability in sensitivity to the changes in sigma factor numbers. Based on standard models of transcription, we have generated the following hypotheses. The binding of a sigma factor to a core RNA polymerase is represented by two chemical reactions on the upper left, one for sigma 70 and the other for any other sigma factor. Importantly, sigma factors compete for a limited amount of core RNA polymerases. Consequently, the RNA polymerases carrying sigma 70 are only a fraction of all RNA polymerases in the cell. This is represented in the next upper right equation. Next, consider that, in general, transcription involves a first rate-limiting step, usually named closed complex formation, followed by another rate-limiting step, the open complex formation. It is after this that elongation starts and an RNA can be produced. These two rate-limiting steps are responsible for almost the entire time interval between consecutive RNA production events in the cell. This is represented in the bottom left equation with the total interval being delta t and the closed complex being tau cc, while the open complex is tau oc. Given this, assume that under a specific stress, the cell increases the number of a sigma factor other than sigma 70. Note that this increase will affect only the tau cc, for example by a factor of s, while the tau oc is independent of these numbers. This results in the bottom right equation, which shows that the degree of change in the time intervals between consecutive RNA productions due to a change in sigma factor numbers other than sigma 70 depends on two variables. One is the degree of change in sigma factor numbers, represented by S. The other is the ratio between tau cc and tau oc. Based on this equation, we plotted two theoretical graphs of the expected change in the duration of time intervals between consecutive RNA productions as a function of each of the two variables. First, when one varies S, note how only if the ratio TCC tau oc is large will changes in S cause significant changes in delta T. From this, we conclude that the initiation kinetics of the promoter affects how much a gene will respond to changes in sigma factors other than sigma 70. Second, when one varies tau cc over tau oc, note how only if S-1 is small will changes in tau cc tau oc affect how much a gene will respond to changes in sigma factors other than sigma 70. The second result is obvious in that it means that the higher the changes in sigma factor numbers, the stronger the effect in gene expression dynamics. We next provide empirical evidence for these predictions. For this, we have used qPCR measurements of the target gene and Western blot measurements of sigma factors and RNA polymerase core enzymes. Also, we used the so-called tau plots to estimate the open complex formation time length from these measurements. Further, to validate the previous measurements, we performed time-lapse microscopy measurements of cells producing RNA molecules that are target for MS2 GFP proteins, which are visible in the upper right figure. As these RNAs are produced, they are bound by 96, more or less, MS2 GFP molecules, which makes them look like fluorescent spots. Then, from movies, one can extract the time intervals between their production events in individual cells. Finally, we can collect distributions of these time intervals and from there produce a model that statistically best fits the data. To validate our initial hypothesis, we need cells that differ in how many sigma factors of a certain sigma factor they have other than sigma 70. We chose to compare cells in the exponential and stationary phases because it's very well known that the sigma 38 numbers significantly change between these two growth phases. The left figure shows the cells used and their growth rates. The two sets are clearly in stationary and exponential growth, respectively. The right figure shows the Western blot measurements of sigma 70, sigma 38, and the core RNA polymerase factors, numbers in wild-type cells. These are called RPOS plus cells, and in deletion mutants for sigma 38, which are RPOS minus cells. Visibly, sigma 38 numbers differ widely between these two growth phases in the wild-type strain alone. To validate our model, we need various promoters with different ratios of tau cc over tau OC to assess if indeed this variable affects the responsiveness to changes in sigma factors other than sigma 70 as the model predicts. To obtain such data, we have measured the kinetics of transcription of several promoters, and in some cases has a function of induction strength, since this factor can also change tau cc over tau OC. Measurements were done by qPCR. From the table, column 3, we see that they all differ widely in tau cc over tau OC. These values were obtained from tau plots for each condition, and they're not shown here. Finally, in column 4, we show the measurements of the degree of change in the rate of RNA production between cells in the stationary and in the exponential phases for each condition. Note how these values are tightly, positively correlated with the values of tau cc, tau OC. In the figure, we show that the empirical data fits well with what the model predicts, given the ratios between tau cc and tau OC. Finally, we conducted one validation using time-lapse microscopy and single-RNA detection techniques. We compared the changes in our production rates with growth phase in the TET-A and BAD promoters, as these were shown the weakest and strongest changes in growth phase respectively. Measurements were done in those RPOS plus and RPOS minus cells. Observe the ratios between mean transcription intervals in the stationary and exponential phase. For the BAD promoter in wild-type cells, this ratio is large, as expected from its large value for tau cc, tau OC. For the TET promoter, it is close to 1, meaning that the change is very weak, as expected from its small tau cc, tau OC. Meanwhile, in deletion mutants, both promoters barely differ in the kinetics between growth phases, as expected because these cells cannot express sigma 38 in the stationary phase. The results are therefore in agreement with the model predictions. In conclusion, under stress, E. coli alters sigma factors numbers as a global gene regulation mechanism, but evidence suggests that not all genes respond similarly to such changes in these numbers. We studied this phenomena and found that, first, the promoter responsiveness to indirect regulation by a sigma factor competition is heavily affected by the durations of the closed and open complex formation. This is made possible by the fact that transcription initiation is a 2 rate-limiting step process, and that sigma factor numbers affect the kinetics of only the first step. As the kinetics of the rate-limiting steps are sequence-dependent and subject to regulation, for example by transcription factors, our results imply that promoter sensitivity to indirect regulation by changes in sigma factor numbers is both evolvable and adaptable to stress conditions. We therefore believe that our findings can assist in understanding the global mechanism of gene regulation in E. coli. Thank you very much.